Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 2.4, membrane permeability. So we've talked about the plasma membrane and the components that make it up, but now we're going to be discussing how the structure of the membrane influences selective permeability, which is also a concept we'll be discussing within this lesson. Specifically focusing on the separation of the internal environment and the external environment, and of course how different molecules can pass through the membrane. And our suggested skill is going to be statistical tests and data analysis. 5D, use data to evaluate a hypothesis or prediction, including rejecting or failing to reject the null hypothesis. So, selective permeability. The cell membrane obviously separates the cell interior from the environment. And the cell membrane's got selective permeability, meaning some things are let in and out of the cell and some are not. And the cell chooses by deciding which kinds of membrane proteins to make, because not everything can just pass through the membrane. A lot of compounds require specific proteins that allow them to pass through the membrane. The cell is able to choose. It is a selective permeability through the membrane. And it's also a semi-permeable membrane. Some things are going to go through even if there aren't any proteins, and some things will not go through. So this is a result of the structure of the cell membrane, of our fluid mosaic model. We're going to have gaps in the membrane, meaning objects of certain sizes can pass through. Because of course, our phospholipid bilayer, our fluid mosaic model, is always flowing. It's never completely solid. It's flowing, and some phospholipids are going to be completely saturated. Some are going to have unsaturated fatty tails, meaning there could be some kinks, and this is going to result in some separation between those phospholipids. Basically, there's going to be a little bit of space where objects of certain size can pass through. Additionally, phospholipids have both charged and uncharged sections. We've got our hydrophilic heads facing the environment, and we also have our hydrophobic fatty acid tails. So only objects of certain polarity can pass freely through the membrane because of this variety in charges. And what's the reason for all this permeability? Because we want to create livable conditions inside the cell through regulating what can go in and out. This is like a barrier between the cell and the outside world, and it allows for livable conditions to be created inside the cell. So let's take a look at some of the effects of membrane permeability. First of all, what can pass through the membrane freely? Meaning it doesn't need any support, it's not going to be turned away. Small nonpolar molecules such as nitrogen, oxygen, and carbon dioxide, and small polar uncharged molecules such as water. And water can really only pass through in small amounts. It can pass freely, but not to the extent that small nonpolar molecules can. And if you notice, these have to be small, because if you're large, you can't fit through the gaps between the phospholipids, and they have to be generally not have a charge. They have to either be nonpolar or they have to be uncharged molecules, because if they do have a charge, then they're not going to be able to make it through the membrane because of the different polarities of the phosphate head and the fatty acid tails. So what cannot pass through the membrane freely? Anything hydrophilic. That's large, such as large polar molecules, and then ions, because they have a specific charge. And these require membrane proteins or embedded channels to allow them to cross the membrane. So if we look at this diagram here, hydrophobic molecules, small nonpolar molecules, such as oxygen, carbon dioxide, steroids, they can pass freely in large amounts. Small uncharged polar molecules, water, glycerol, urea, ethanol, they can pass freely, but in less amounts. The area is skinnier. And then large uncharged polar molecules such as glucose and sucrose, because of their size, they can't pass through, and then ions can't pass through because of their charge. So if you're large and if you're charged, then you're not making it freely through the membrane. You need a membrane protein. And finally, let's take a look at the cell wall and how it affects cellular transport. So cell walls for plants and fungi are made out of complex carbohydrates. In plants, cellulose. And they're used for structural support. They provide a structural boundary, so they help keep the shape of the cell. They help provide another boundary besides the membrane, and they can also help maintain a barrier between internal and external environments. So over here we have a plant cell. It's got the cell wall on the outside. We can see our plasmodesmata. These are the pores that things are going to be able to pass through through the cell wall, then cross the membrane and get into the cell. And obviously, these are going to block large molecules. So the cell wall is like an extra level of protection. It's another barrier, and it's going to block large molecules from passing through. The charge doesn't matter as much here, but size is still going to matter when we're looking at what can pass through the cell wall. So that's it for membrane permeability. Here's the review for topic 2.5. You want to know about selective permeability. How does it result from the structure of the cell membrane? What is it going to result in terms of cellular transport? What can easily pass through the membrane? What can freely pass through? And what cannot freely pass through the membrane and requires proteins? And then also how the cell wall is going to affect the cell function and membrane transport. So next up, we're going to talk about the different kinds of transport through the cell membrane. But for now, hope this was helpful.